When we were counting up ATPs a couple of days ago, and we counted up just the ATPs you made in the far right-hand column from all the NADH2s and FADH2s that you took through Krebs cycle and electron transport system, it was 34 ATPs that you made in just those 2 steps. The other 6 gross ATPs were made right in the pathways of glycolysis or when you were going around Krebs by itself. But the ones made from NADH2 and FADH2 ended up becoming a gross of 34 ATP. So that's what this question 22 is talking about, is those 2 steps when you brought all the NADH2s and FADH2s that were so many that were made in Krebs and then brought down into the electron transport system, and any NADH2s or FADH2s that came earlier. So I just want you to understand why they gave that area a special name. What does phosphorylating something mean you're doing to it? Adding phosphate groups to it. Who are you doing that to in this story? ADP. So that's what the phosphorylation means. It means you're taking an ADP and you're adding an inorganic phosphate to it to accomplish making your ATP that you wanted, okay? Oxidation. I told you what oxidation reduction reactions are. I used the example of the electron transport system where you had compound, cytochrome compound A, B, or C. And I said, you off your NADH2 or FADH2, you knocked hydrogens off and they'd come over here and bond. That part of the reaction was called reduction. Well, the important part of the reaction is when this goes zzz and busts that bond. And now that got removed off of the first cytochrome compound. Removing hydrogens and electrons is called oxidation. When you busted that bond, that's the energy that allowed you to put this on this to make that. Are you getting the picture? So it's called, the oxidation half of the reaction is the important half, not joining the hydrogen there, but breaking the hydrogen off. So oxidation gives you the energy to do the phosphorylation. So those 2 steps together are called oxidative phosphorylation. Is there anybody who doesn't understand where you are in the pathways? Okay, well, moving on then through the rest of this study guide. This is your study guide for metabolism. When finally the hydrogens let go of NADH2 and FADH2 and the cytochrome compounds ABC, who's the final hydrogen acceptor? Oxygen. Oxygen. And what does it end up forming that you exhale? Water. What's the only other thing you exhale? CO2. Okay. Now the food group that you're using is protein. If you're using protein, you hack off 2 non-usable ends. If you hack off this end and throw it away, then removing that end is called what? What's this group called? Deamination. On the other end, if you hack off this group, what is that called? Decarboxylation. And that leaves you a usable chain of carbons down the middle depending on which of the 20 amino acids in your diet you're talking about. So you could have become a 2-carbon group, a 3-carbon group, a 4-carbon group, or a 5-carbon group. If you are a 2-carbon group, what they're asking you is where do you fit into the aerobic respiration pathways? As what molecule? What's the only 2-carbon group in there? Acetic acid, which is in what step? At the end of bridge transition. Speak up just a little bit more, because I'm old. Okay? So that's what they're asking you. And any ATPs or NADH2s or FADH2s that you could make past that point, then this protein scrap gets to make. Where does the 3-carbon protein scrap fit in? Yes, pyruvate or pyruvic acid. If you're pyruvate, where are you? You're at the end of glycolysis, so you go forward in the pathways from there and make all the energy you can. If you're a 4 or 5 carbon compound, what step in the pathway are you going to fit into? Krebs cycle. What is the 4 carbon compound in Krebs that we learned? Yep. Oxaloacetic acid. And what's the 5 carbon compound we learned? Yes. Alpha, keto, I couldn't get it all on there, glutaric, and then acid. Alpha ketoglutarate or alpha ketogluteric acid. This is alpha ketogluteric acid. Can you reverse the food scraps in the metabolic pathways and remake a protein? No. What do you have to do? How do you make a protein? What? Transcription of RNA off of DNA, and then you have to use the RNA to haul the amino acids over to the ribosome structures to assemble the amino acids in the right order to build a protein, and all of that business is called translation. So you've got to do transcription and translation. Okay, are you caught up with me? Now here you can either put the number up here by the answer, or however you want to do it, put the letter down here, but what is the vocabulary word for making a new glucose out of food scraps left in the pathway? Gluconeogenesis, number 3. What do you call the abnormal condition? Because you should never have 1 molecule of glucose in your urine. So if you have detectable glucose in your urine, what do you call that? Glycosuria. That's one of those URIA words, number 1. What is the vocabulary word for you are making glycogen? Glycogenesis, number 5. Genesis means to make or create. What if you're breaking down glycogen starch? Yeah, glycogenolysis. Number 2. Lysis means to rupture or break down. So that's number 2. What if you are making a fat? Lipogenesis, number 6. Where's the only place you can finally make that fat? Couldn't hear y'all. In your adipose cells, adipocytes. But if you want to break down fat, you have to mobilize the fat out of your adipocytes and take it to your liver to break it down. And then you can send it out and start using it. Where is more than 50% of your fat stored? I think somebody over there said it. In the subcutaneous layer or hypodermis layer of your skin as an insulating blanket. If you were going to pull phosphoglyceraldehyde out of the pathways, what food scrap does it resemble? Glycerol. Glycerol of a fat. The building block of a fat. What can be put together to make fatty acids? Acetic acid. 2 carbon acetic acids are strung together to make fatty acids, and they're also strung together to make this thing. What is this thing? This is a cholesterol. 2 carbon acetic acids are strung together 2 by 2 to build that in your liver as well. But anyway, this said we're making a fatty acid, not a cholesterol. If you're going to catabolize or break down a triglyceride, what is the vocabulary word for that? Lipolysis. Lipolysis. And beta oxidation creates mainly ketone bodies, but a little bit of this. What is this? Acetic acid. So you used acetic acid twice. You used it there and in another one. You remember beta oxidation was just the fatty acid chain, that every 2 carbons you were breaking it down, and most of the fatty acid becomes bad things that your liver can't handle. Ketone bodies, that's why we say you're in ketosis, a condition of ketone bodies, or ketogenesis, you're generating ketone bodies. But then your liver sends those out to your cells, and your cell can turn those 2 carbon compounds into acetic acid. An example of a toxic ketone body is acetone. Okay. Burning fats instead of sugars causes a condition called condition. What's the suffix that means the condition? Osis. So what condition? Ketosis. And due to waste from what part did you just break down to create those? What part of the fat? What part of the lipid? Be more specific. What are the 2 building blocks that make a lipid or a fat? Fatty acid and glycerol. So which one is it that just created these 2 carbon wastes? The fatty acid. So this is a fatty acid catabolism problem when you end up with ketosis. You've been burning fat, yes, but now you're using this half of fat. That's the half that gives you trouble. And it makes somebody acidic as well, because the fatty acids tend to let go that last hydrogen. All right, give the proper term for each. So we're talking about temperature control mechanisms. Heat is moving from a warmer to a cooler object, and they are touching. Conduction. Heat is moving from warmer to cooler object through air or water. Convection, like an oven. Convection oven. Heat is moving from a warmer object and travels to a cooler object, but they're not touching. Yeah, and what the thing to say about radiation this is the biggest heat loss mechanism in the human body, is radiation. This is a water gradient, an osmotic gradient, which carries heat off the body to the environment, because water is moving from your body that has a lot of water to air that is drier. Yeah, that's evaporation. And the claim to fame about evaporation is that's the only mechanism that can cool you off if the environmental air is hotter than your body is. Because the top A, B, and C won't work when the air is hotter than you. In fact, you'll be heating up more and more due to the fact that the environment's hotter than you. Okay. What's the vitamin for night vision? A. Beta carotene. Necessary for calcium absorption, because it's the carrier molecule. D. Which one in blood did we study for the production of prothrombin, or you'll have clotting problems in 24 to 48 hours? K. Which one's required for red blood cell formation, and it's part of NAD and FAD? In here it's niacin, and here it's riboflavin. Yeah, B complex, B. So that leaves which 2 to be for general healing and health of body membranes? Yes. So a lot of people who are going into surgery or who have been ill comment on getting A and E and C, because they feel like they promote healing and keeping membranes and capillaries healthy. And these 2 are antioxidants, which means they're scavengers who run around and look for something that got in your body in a bad form. This one's for general good growth. Okay, that's a review of your metabolism information. Certainly you should learn all the notes that we covered on that, but don't lose that. Study that significantly. I'll say again, this is not part of this Monday's digestive test. This is not digestion. This Monday's test ended after you finished with hormones like enterogastrone. You're getting your lab test in lab, by the way. The other half of metabolism is urinary, because you got to deal with all the waste you generated. Do I need to give you one more of anything else yet? Study guide? Philip?
It doesn't matter.
Okay, did she get a study guide? No. Is there anybody else I forgot that y'all told me is ill? It's definitely going around. Clorox bleach is your friend. Now, you notice the first thing it says. Review the structural notes from lab. So that's the big anatomy thing that we had. We actually had 2 labs you need to review. The one that did the major and minor calyces and the blood vessels through the kidney, you need to review all that prior to this lecture test. And you need to review the urinalysis sheet that I gave you. The 2 left-hand columns particularly, where it said these are the characteristics you have to look for in urine, like pH and color and odor and so on. And then the second column that told you the norms, like if it said what wastes are in urine, then it listed the ammonia and so forth. So, and it also told you to know all the URIA words like pyuria, hematuria, ketonuria. So know that. So have you clarified that so you know what you're reviewing? If I gave you any special vocabulary words like position of the ureter and kidneys is retroperitoneal, and the name of a kidney stone is a renal calculus, any of those vocabulary words that I scribbled onto those sheets you should know. So review what we went over. What I don't expect you to know is how to diagnose somebody. That right-hand column that told you some of the abnormalities, like things that discolor urine and things like that, like tumors and stuff. You don't have to memorize all of that. Those were just examples to show you how understanding the norm could help you realize when something wasn't normal. All right. Know the organs. This is a fairly simplistic organ system. 2 kidneys usually. There are people that have just one centralized kidney that never divided, and they're perfectly normal. You can live fine with 1 kidney. 2 ureters to drain the 2 kidneys, to your bladder, and that bladder, if this is your bladder, the ureters come down and dump into your bladder, and then you leave the bladder through an orifice to your urethra. And so that creates this little triangle region right here in your bladder where you triangulate the 3 orifices. That is known as the trigon region in the bladder. I've gone over these major functions of your urinary system. A biggie is getting rid of nitrogen-containing waste, which are called nitrogenous waste. And many of them come from breaking down proteins, but there are nitrogens in your DNA and RNA as well. So those are the 2 big food groups that they come from. Shoot, I had one I wrote all over now. So these are some of the things I said before. Urea was made by your liver, and when it gets to your bladder, every trip around, half of it is excreted out of your body from your bladder. That's not true. Half of it is excreted from the blood to your urinary system. Which is to say, you'll be getting rid of half of whatever quantity of urea that you have in your bloodstream every time you go past your urinary system. And then the next time you go by your urinary system, you'll get rid of half of whatever you have in your system at that time. So it varies how much you're dumping into your urinary system dependent on how much of it there is in the blood. It's a diffusion thing, obviously. That's why it's always in equilibrium. You're only getting rid of half of whatever's in there. Ammonium, as we say, a lot of these are clipped off of the amino acids that build proteins. The nitrogen-containing compounds from your DNA and RNA are called uric acid, so you're getting rid of that. This is a nitrogen-containing waste. It is called creatinine. It is the waste that is what a storage molecule in muscle and nerve tissue called creatine phosphate becomes when it deteriorates. So this is just telling you the usual location of this ATP storage molecule, which was known as creatine phosphate. When it deteriorates, it becomes this bad compound, creatinine. So this was the last depot or storage site for ATP in your muscle and nerve tissue. The first place you stored ATP was on myosin bridges in muscle, but then when you got them all loaded up, you go load your creatine up with it and make creatine phosphate. Any question on that part? We had been through that once already. Certainly conserving water is an important role of your urinary tract. We need the water back in your body so that you can maintain normal blood pressure and you don't have a low blood pressure. And if there's excess salt or other solutes, then that is going to have an even bigger effect, because if there's excess salt in your blood, that's going to draw more water to your blood. Therefore, the next place it's going is your urinary tract. So one problem leads to another. pH balance, somewhere between 6 and 6.2 is the average pH of urine. Mainly it's down there in the acid range, 5.5. It did come up to 7.5, so it will enter the alkaline range and be normal, but this is just the average. If you have a fever, that tends to make your pH lower. If you're on a protein diet, that tends to make your pH lower. If you're on a very vegetarian diet, that tends to make the pH higher. If you're retaining urine in your bladder, that tends to make the pH higher. And this is just meant to be an excretory waste that's going out of your body. So if there are valuable foods or things that are needed in the body, your kidney has the ability to recycle good things back into your body bloodstream. So these are some of the things that you would recycle if you needed them. This you would recycle 100% of. This you would recycle 100% of in a normal, healthy individual. These you'll recycle back to your bloodstream if you're needing them at that moment in time. If they're excessive in your body at that moment in time and you don't need them, then a lot of minerals and vitamins are lost in your urine. about the only vitamins that are typically lost are water-soluble vitamins. So that would be your C and your B complex group. Most of the fatty vitamins, vitalipids, wouldn't. But all kinds of minerals that we went over before, sulfates, phosphates, calcium, potassium, some sodium, some chloride, and I'm sure there's tons of others that are not on the list. But that's why we tested for some of those things to prove they could be normal constituents found in urine. Okay, pretty much that's been a review of where we've been before. Now this is a little picture of a nephron dumping into a collecting tubule, but if you don't like it, draw yourself a quickie one right here. So I have highlighted in yellow the parts that make up a nephron. You are a nephron from the glomerulus up to, but not including, the collecting tubule. That's not part of it. A collecting tubule collects from many different nephrons. If you left this glomerulus capillary bed out of the story, then the remaining yellow portion is called renal tubule. So that's what I have written up here. That's why it's important that you know that you have to include the glomerulus, because otherwise your textbook is referring to it with different terminology. So we're fixing to talk about the 3 steps that your body does to make urine. And it runs these steps, and it says step 1, and there'll be a step 2 and a step 3. It does these 3 steps simultaneously. I'm just breaking them apart so you understand what's going on in these 3 steps. And you aren't urine until you leave the collecting duct, because anywhere up to that point, you could get soaked back into the human body and not excrete it. Urine's an excretion, leaving the body. That's what excrete means. But anything prior to that point, your body could go, no, I changed my mind, give me that back. So you only call it urine once it's made it this far. So prior to that, you call it filtrate. So in step number 1, it is called glomerular filtration. And it's going to be a mass loss of filtrate from your blood dumped into the glomerulus capillary bed, and from there it's going to sieve over into Bowman's capsule. So in other words, step 1 is happening right here. That's step 1. Your waste and a lot of fluid are leaving the glomerulus and going into the nephron. And you call that stuff that now is in the tubules of the nephron filtrate. This is a good thing, Martha Stewart, that we're having this mass blood out of your bloodstream, because in this big gush of fluid, you will take out lots of waste. Lots of things you're wanting to get rid of. The bad news is you also lost a lot of things you never intend to lose. So there'll be step 2 where you go, no, I want that back. So this is just a mass movement in order to get rid of about a bunch of really toxic substances. So there are factors that aid glomerular filtration occurring so well. One of it is, you saw one little plane in lab cut through a kidney. on a green plaque model, and all the little dots out in the cortex, each were a glomerulus capillary bed. And there were several dozens just in one little area on that one plane. And you'll have plane after plane after plane through a kidney. So there are literally thousands and thousands and thousands and thousands of glomerulus capillary beds in your two kidneys. So if you took the surface area, cut the capillaries open and spread it out and made a carpet out of all the surface area of just glomerular capillaries, it's equal to the surface of the whole human body. That is a lot of surface area over which to move filtrate. So that's number one. Number two, compared to any other capillaries we know of in the human body, these glomerular capillaries are 100 times more porous than any other typical capillary beds. So things are going to sieve out of them very easily. And number three, it comes down to high to low pressure. So let's see what the numbers are. There is a pressure operating in this direction because you've got pressure in the bloodstream. And so that is going to be called blood pressure. And the blood pressure is about 75 millimeters of mercury out of your bloodstream and into the nephron. Blood pressure. You have it right here. But there's particles too big to get out of the blood. We've talked about this before. Red blood cells don't get to leave. Big proteins don't get to leave. Minerals don't all leave. Those are called solutes. Solutes act like a sponge or a wick. What do they attract? What do they attract? Water. So if those are left here, you have got a force that's trying to attract things to go back in there, the solutes that were too big to leave. And that is called the osmotic pressure of blood. So that's OP, due to things, solutes trapped in your blood. You've got that discussed right there. But the key thing is things trapped in your blood. And then, if you sieved all this stuff over here, like filling up a water balloon, then this Bowman's capsule or glomerular capsule is bloated with filtrate. Does it want to take on any more filtrate? No. So it is pushing back because it is filled with filtrate. That's called capsular pressure. So that's an outward, out of the nephron force, back in this direction. That's CP, capsular pressure. So you've got two forces trying to resist filtrate coming into the nephron, and one force, blood pressure, trying to push filtrate into the nephron. So it's all a math equation to see if these two pushing back into the blood are bigger, or whether this one pushing out of the blood to fill the nephron is bigger. And so here's the numbers. Osmotic pressure was 30, capsular pressure was 20. So this is blood pressure, 75. This is capsular pressure, 20. This is osmotic pressure, 30. So these were the two that were pushing back to the blood. This is the one that was pushing out of the blood to the nephron. So it's 75 minus 50. And so this is the force with which things will exit your bloodstream to enter the nephron and form filtrate. And that's called your effective filtration pressure. So it's abbreviated P sub EFF. It means effective filtration pressure. They can measure how much filtrate is made in the human body in a minute, and they have, and so in an adult, it's 125 mLs of filtrate made every minute, and that's called your GFR, your glomerular filtration rate. that is equal to 1/5 of the renal blood is going over into the nephron and could end up in the bladder if it stays there. Which is to say 5 trips around and all the blood going to the urine, those vessels would be empty if we weren't going, hold it, I want some of that back. So let's see what that means in a 24-hour period. If this is how much per minute, this is your how much per minute filtrate you're making, and you multiply that by 60 minutes in 1 hour, and you multiply that by their 24 hours in a day, then the minutes will disappear and the hours will disappear, and you'll end up with mLs of filtrate in a day. And if you did your handy dandy calculator and you multiplied the 125 times 60 times 24, the answer comes out you make 180,000 mLs of filtrate in 1 day. Who cares? Well, if that's how much you make in a day, you know how much urine from the other day's lab you make in a day. 14 to 1,500 mLs in a day, remember? What would you have to divide this by to get anywhere close to 1,500? You'd have to lose 2 zeros, right? That's 10 times 10. You'd have to divide this by 100. What I'm trying to say to you is, even if 99% of this was reabsorbed, that would still give us more urine than we actually eliminate in a day. So we're going to have to reabsorb more than 99% of this. And less than 1% of it will actually end up being urine. But it was still a good thing that we did this big flood, because in this big volume of liquid, we hauled out a bunch of toxic waste into your tubules of your kidney. And that's the point of this step. The rest of the steps will be much more selective. But because they are, guess what? It's going to cost you some ATP. This didn't cost you anything but a pressure gradient operating. Are you with me so far? Okay. Step 2. What is this called? Afferent arteriole. So then what is this littler one? Efferent arteriole. And so they do that on purpose. They make it skinnier on purpose so that your blood pressure never changes from here to here. By being a smaller diameter, even though you've lost a great deal of fluid, you're keeping your blood pressure safe, at safe levels. But because you came out in an arteriole, this arteriole has to break down into a new capillary bed. So what is the name of this capillary bed? Peritubular capillary bed. Now, if you're a meticulous reader, there is a certain part of this capillary bed that is called the vasa recti, but the whole capillary bed is the peritubular capillary bed, and that's all I'm ever going to test you on is peritubular. I'm not going to go to specific regions of it. But, so you got your little picture here going. The reason I made you redraw the picture is where this step is going on is from here, your waste in a nephron, and you're getting kicked out of the nephron and put back into the bloodstream. So you're going that way, back into the peritubular capillary. So this is where step 2 is happening, and in that direction. Okay. Number one, 180,000 mLs of filtrate. We need over 99% of that water back in our body, or our blood pressure will go boom. So we got to retrieve water, we know that. 100% of your glucose sugar got kicked out of your bloodstream and is out there in filtrate that could become future urine. Is there supposed to be any glucose in urine? So we got to get all the glucose back. And there are a number of other solutes that got dumped out there, like amino acids, for instance, and certain minerals that we want to get back. So this is the one and only step where you get back stuff that's valuable to your body. So you're putting it back where it was before. That's why it's called reabsorption. So it's tubular reabsorption because it's from the renal tubules back into the capillary bed. That's the name of step 2. It's the only step where you get stuff back. And so this is just verbally telling you where it's occurring. And here's examples of some of the stuff your body wants back. Now, in step 1, 100% of your glucose got kicked out. In step 2, right here, we're hoping that 100% of the glucose gets picked back up. When you have it, if you're going into allied health care area like nursing, and if you were on a renal ward, they try to figure out if somebody's in there with kidney malfunction, which step in urine formation is your kidney not doing properly, because they either want to correct your disorder and get your kidney to pick back up and do what it's supposed to. Otherwise, they've got to create a dialysis bath that will mimic the step you're not doing right so that it'll move things for you. Do you think we're going to be able to do it as good as the human body? No. So anybody on renal dialysis is compromised because you're leaving something behind you shouldn't have, or you're taking too much of something out of the body you shouldn't have. But it's the closest mankind can do to mimicking the physiology of the human body. So what they do to test you to see which step you're screwing up on, for instance, if they wanted to see if you were screwing up on test 2, they know that you should not, at the end of this step, have any glucose left in your nephron, because it should have all been put back into your blood at the peritubular capillaries. So they do what is called a renal clearance test with the metabolite, in this case the metabolite being glucose, that should have all gotten thrown back into the blood at the end of this step. And so what they'll do is they'll give you a super dose of glucose. They sometimes call that glucola. There's several names to that sugary drink. Sometimes when you're a pregnant mom, they make you take this test once during your pregnancy, because some people go into diabetes mellitus just during their pregnancy, and about the time they give birth, they're not having that problem anymore. So it's just one of those things they watch when you have that excess 20 pounds, 70 pounds out there. But anyway, so if they give you this X amount of sugary drink, and they know that they've given you more than you can possibly pick up, then they know what percentage should show up the first time you urinate in your urine. And if that right amount shows up in your urine, they know that your step 2 worked just fine, because they deliberately overloaded you with more than you could possibly pick up. But if you show up in your urine with far more than you should have been eliminating because of the prescription of this drink they just ordered for you, then they know that you didn't do step 2 right and pick up your glucose properly. So they know that that's one of the failing areas of your kidney, is tubular reabsorption. And so they'll pick a different metabolite or mineral or whatever in each of the various steps to prove which thing you're not taking care of properly. Okay? That's what's called renal clearance tests. So let's look at what's going on here. There's two kinds of transport mechanisms you learned in Anatomy 1. One group was called passive transport because it doesn't use a single bit of ATP. But there are certain rules that have to apply if it's not going to cost you ATP. You have to be going from a high to low gradient. Now there's different kinds of gradients. You could be going from a high to low concentration gradient, like a lot of water here, not much water there. That would be a concentration gradient. You could be going from a high to low pressure. We saw that in step 1. Pressure drove you out of the glomerulus capillary. Or you could be going according to electrical gradient. A lot of something positive moves, so who came for free right behind it? A lot of something negative, because opposites attract. So there are different kinds of gradients, but it has to go high to low, whatever the gradient is. Now, this accounts for, remember we were going to reabsorb more than 99% of the water that was lost from your blood in step 1? 99? 80% of the water will get put back into the peritubular capillaries and therefore retained in your body just because of the solutes that were over there in your blood. Solutes act as a wick. They attract water. So if 99% of your water right now is over here in your nephron, the solutes over here in the blood are going to attract it by simple osmosis. It'll move back into your bloodstream. Well, that ain't enough. How much more do we need? At least 19% more or 19.5% more water we need to reabsorb, or we're eliminating too much body fluid. So this is obligated to move. It has to move due to the solute concentration in the bloodstream. This is called optional, facultative water reabsorption. If you're in microbiology class, you have facultative organisms. Those are organisms with options of how they can live. They can live without oxygen and not die, or it's an organism that can also live with oxygen and not die. Not all organisms are like that, but an optional one called facultative can do different things and it won't hurt them. All right, well, what makes this optional for us is if you secrete a hormone, you get the extra 19.5% water back. If you don't secrete the hormone, then that option is now you don't get that water back. So you're counting on your glands secreting the hormone. The hormone is a steroid, therefore it ends with the suffix sterone, and it's called aldosterone. And in a nutshell, it makes you save sodium chloride, salt. Well, what does salt do? Draws water. So if you save extra salt, you're going to get extra water back into your system. Because you got extra water back into your system, what does that do to your blood pressure? It keeps your blood pressure up. So this is a blood pressure maintenance thing as well. So you don't bottom out. And the 1% or 0.5% filtrate that never got taken back out of the nephron and put in the peritubular capillaries, that's the amount that will be the urine. And it makes the 1,400 or 1,500 mLs of urine per day. Urea is passive because it's just going to diffuse out of the nephron into the peritubular capillaries. 50% of it will remain in the nephron, 50% of it will go into the peritubular capillaries. So that's just an equilibrium, 50-50 diffusion story. Now, all those important foods, glucose, amino acids, some important ions, those are going to cost you some ATP bucks. You're not getting them for free. You want them bad enough, you're going to use ATP. So the most abundant cation in our extracellular fluids, like urine, is sodium. Therefore, the most abundant anion, negative thing, is its partner, chloride. They're usually there in equal amounts. So the positive thing is cation, and the negative thing is called an anion. I remember it because it has two N's in it, so it's the negative thing. N, negative, two N's, anion. But anyway, when you're looking at this nephron, if you were filtrate, you're moving right now in this direction. So this side of the loop of Henle is called the descending limb of the loop of Henle. But then when you get over here, you're now going this way through the loop of Henle. And if you're going this way, then this is called the ascending limb of the loop of Henle. And you know, folks, I know you're getting tired at this point in the semester, but let me tell you what. They will give you little quizzes like this in nursing now. They have gotten so sick of people that have been through anatomy 1 and 2 going down there and they don't even know the route of blood flow through the heart and what the valves of the heart are called and what the sinoatrial node and the AV node and the bundle branches and all that. So now that in pharmacology, they give them a test on that nervous tissue, that conductive tissue of the heart. They give them a test on the valves of the heart, the chambers of the heart, and the path of blood through the heart to the lung and back to the heart. So then when you get to the renal section, they'll hit you up on this. So you may think I'm just picking at you, but you're going to see it if that's where you're headed. So just know that. Don't lose this stuff. But anywho, so the deal is this. If we're trying to get some important minerals back, our most abundant ones, then you're going to use carrier molecules. Now this is molecules, and therefore that requires some ATP to make sodium go back into the blood in this descending limb. So sodium's moved first. So sodium would go this way, and that'll be number 1. And then because you put all the positives back, then according to the electrical gradient, you will move the chloride for free. No ATP required. But on this one, ATP is used because you had to force the sodium to move first. So you're going to get that moving first, and then secondarily, you're going to get the chloride as the number 2 ion moving back into the blood. That is true everywhere in the nephron except right here. In the ascending limb, it's just the opposite. You're going to move chloride first, and then the sodium is going to be second. But it's the same rule that whoever is moved first, you have to force it to move by using ATP. Whoever moves second just does the electrical gradient and is attracted to the other charge. So that's how you get minerals back, is using a carrier molecule. And usually, from Anatomy 1, you probably learned this, but to get something to bond to a carrier molecule, you usually have to add a phosphate group to the carrier molecule. That's phosphorylating it. That's why to hook something to a carrier, it costs you ATP, because you had to change the carrier molecule. It's a rare instance when something is naturally attracted to a carrier and you don't have to alter it in some way to make these things happen. Okay, so it summarizes that for you verbally there, but that's where that is happening. We're still trying to get back stuff we want. We want the glucose back. So we've shown you how you get some ions back. Here's the deal with glucose. If the norms that they allow you to have in your bloodstream are 80 to 125 milligrams percent glucose for every 100 mLs of plasma in your blood, then that's how much you can have and not have too much. Actually, your nutritionist and your doctor, just like cholesterol and all the good stuff, they're getting stricter. So your doctor may want this upper number now at 100. So a number might be more like 70 to 100. But even then, what I'm fixing to teach you will still apply. But you'll have to ask your doctor, well, what do you think? Because they all don't They fight about what your cholesterol can be these days. They fight about what your glucose can be. So you go with who you trust medically. But anyway, here's the deal. To get that solute back, it's a huge molecule, glucose. You're going to need a carrier molecule to carry it back into the peritubular capillaries. So the number of glucose molecules you can get is going to depend on how many carrier molecules for glucose you have in your bloodstream. So when you flip the page, you'll see how many you have. Carrier molecule availability is your tubular maximum. That's the maximum amount of glucose that you can grab and get back in your blood. So it's abbreviated your T sub max. So your T sub max for glucose is 160 to 180 carrier molecules. If the amount of glucose you have in the filtrate is 80 to 125 glucose milligrams, should you be leaving any behind? In your filtrate, your future urine, you have 80 up to 125 if you're normal. This is how many carrier molecules you have to get it back. Should you be leaving any in the nephron? No, you got way more carrier molecules than the amount of glucose that's supposed to be in there in the first place. That's why we should never see a glucose molecule in our urine, because we have far more carrier molecules than you have glucose that's supposed to be showing up. Okay? So 100% of it should get returned to your bloodstream. Now, critical thinking wise, insulin's the hormone that's supposed to take glucose out of your bloodstream and give it into your body cells. Let's just say you're not making insulin. So it didn't take some of your glucose out of your bloodstream and put it in your body cells. Then what's happening to the glucose level in your bloodstream? It's going up and up and up and up and up, and the minute it gets above 180, will you have carrier molecules to pick it up? And no. And that's when it shows up in your urine. That's why that's a sign of sugar diabetes, diabetes mellitus, because you didn't have enough insulin to keep the glucose down within the normal level in the bloodstream, so it crept up and up and up, and pretty soon it got higher than the carrier molecule concentration, and that's when it shows up in your urine. See where all this fits in now? Okay. Just like I just showed you on sugar, on glucose, that's how you get an amino acid back, or sodium or potassium, if you're wanting it. But it wouldn't be the same carrier molecule, nor would the numbers be the same. But you'd have to have your normal carrier molecules for each of these valuable metabolites in order to get them back into your bloodstream. But they work just like the principle by which glucose worked. Okay. Step 3. Step 3 is at the same location, but we're not putting stuff back in your body blood. Your body blood is saying, ooh, I still have some bad things in my bloodstream I would like to dump. So step 3 is going this direction, out of the blood and dumping back into the nephron. So this is step number 3. It is between the nephron and the peritubular capillaries, but we're dumping it out of your blood and giving it to the kidneys tubules. So that's got to be some really bad stuff. And it's going to cost you carrier molecules, and therefore it's going to use some ATP. So it's very similar to what we just saw. We're just going the opposite direction. Carrier molecules and ATP is used. So we might want to get rid of excess acid, hydrogens, ammonia, ammonium. What would we say your urinary system is acting as if we're getting rid of this, and 3 hydrogens there, and 4 hydrogens there, and 4 on urea? It's a major buffer mechanism, your urinary excretion, because you're getting rid of all of these potential things that could affect your pH. So that's a major buffering role that your urinary system provides. You could get rid of excess potassium unless you needed it, and then you can release hormones that make you retrieve it. We'll get to those later. There's that nitrogen-containing waste from ATP storage, creatinine, drugs. This is why urinalysis has become a tool to see if someone's cheating, like in sports events. Because if you have taken some drug, then your body doesn't recognize that as normal, and it will kick anything excessive over the normal amount out of your body as part of urine. So we can tell if you've taken steroids like testosterones. Even if you took them a couple of years ago, they're not usually in excess in your body. You only usually make the amount you need, so they're not sitting around being stored. So if you've taken excess amounts, it is a fat-soluble compound, your body will store it. And even if it doesn't store it exactly as testosterone, they've analyzed it to figure out what it would be broken down into, and they can still prove that you took something in an artificial dose. So that's urinalysis. Marijuana urinalysis, same deal. They know what compounds they're looking for. It shows up in your urine, and on you go with that kind of thing. So we said this, getting rid of number 1 and number 2 and number 4 helps balance pH so that you don't become too acidic. If you had the opposite problem and you were becoming too alkaline, you can attract with this potassium OH minuses and get those to go out of the system. So the urinary tract helps you on either end of a pH imbalance, alkalosis or acidosis. We said in lab, don't say pee or piss. The two most accepted terms are urination and micturition, mean excreting urine from the body. Void would probably be acceptable, but if you're in the clinical environment, pay close attention to what terminology they use for things like that. This is a reflex, just like defecation is a spinal reflex, and it actually uses the same exact nerves. What happens is, as your urine distends, stretches with mLs of urine accumulating, you have pressure receptors in the wall of the bladder. So when you have 200 to 400 mLs or cc of urine in your bladder, you get the urge that maybe it's about time to eliminate. But maybe you're stuck in traffic after a Rays game, and all of those pretzels and beer, and the bridge is closed because some crane's about to fall off a barge. Did you see that today? And so they won't let you over the bridge. And so maximum capacity for most people's bladder is 400 to 600 mLs or cc's of urine. And so finally you got to the Raceway gas station and lickety split, there you go inside. Otherwise the urine is backing up in your ureters to your kidney, or you're going to urinate involuntarily, one or the other. So that's the maximum capacity of most adult bladders. So to remind you, if we're not potty trained or we're allowing urination to occur, it is a parasympathetic nervous system reflex. So a nerve is arriving at the spinal region of your vertebral column. That's an afferent neuron. It's arriving there. There's a connecting neuron in that area. In Anatomy 1, it was called an association neuron, or it was called an internuntial neuron. Two names for the same thing. The point is, that's in the vertebral column, and it hooks you up with the exiting parasympathetic neuron. That's an efferent neuron. So this is just a chain reaction. Once it starts, you can't stop it. When it comes out of the vertebral column, it hits a pelvic nerve. And the pelvic nerve is part of the involuntary nervous system, and it is stimulated when you're running the parasympathetic side, which when you're talking urinary system, that is the side you're doing. which means if you're running this one and firing it, you are inhibiting the sympathetic nervous system and not stimulating it. So this parasympathetic neuron will not fire to your muscle, which is called the internal urethral sphincter. If you're not firing to it, it's going around your urethra. It is not constricting. Therefore, it has relaxed, and that allows the urine to enter your urethra. At the same time that pelvic nerve is relaxing the internal urethral sphincter, it did fire to the bladder muscle. The bladder muscle is called the detrusor muscle, and it's involuntary smooth muscle, and it contracts to propel the urine out of that storage bladder into the urethra. So we got two things going. This is contracting, it's pushing urine into the urethra. The sphincter on the urethra is open. Now that just leaves one more urethra, the external urethral sphincter. That same parasympathetic involuntary nerve inhibits a voluntary nerve. This is the one voluntary step. This is called the pudendal nerve, but it inhibits this so that it doesn't fire. Therefore, the external urethral sphincter, which is made of voluntary skeletal muscle, doesn't constrict, and therefore it's relaxed, and therefore urine can go on out of the body. So this is your last doorway. This is on the outside of your perineum, your pelvic floor. So that's what happens. If you're potty trained and you go, nope, I'm stopped at the bridge, now's not a good time, the only way you can prevent this from relaxing is if the conscious center of your brain, the cerebral cortex, fires, then that will stimulate the pudendal nerve, and then and only then it fires. And if it fires, it's going to constrict that urethral sphincter. So this is the only side to get you to constrict that. So if you need to clarify that and say, when this fires, the sphincter is going to constrict, then do so. Otherwise, all sphincters will be inhibited and relaxed. Okay, we'll stop there. We're not done with the urinary system. There are two big hormone pathways that control your urinary system, so we will be talking about those. But when you come on Monday, you're having your digestive lecture test. Do not start cutting tests